Hi. Now let's discuss about the biological responses to biomaterials, especially the foreign body response to implanted biomaterials. So you can consider that some of the implants, such as, let's say, breast implants into the breast of a human, and that silicone, it depends on the person and it's still not yet clear. But over time, these biomaterials, our body considers as a foreign body. So try to engulf if it's very small, but in general, these biomaterials are big. Then our macrophage, which is our immune cell, the cell-eating cells, try to engulf it, but it's impossible. So it's frustrated. Later on, they secrete, also generate a collagen secretion by fibroblasts to make a collagen capsule to effectively consider this as a foreign, even if it's inside our body. So it causes a problem, inflammation, and later there could be a serious problem can happen. Okay. So implanted biomaterials, there are complex biological response to an implanted biomaterial due to the presence of our body, the blood, and inside tissue, interstitial fluids, and also multiple cell types, which will directly or indirectly interact with the implanted biomaterials. So what are typical responses? So first, proteins from the blood plasma and interstitial fluid will directly touch and coat the surfaces of biomaterial. And that we call as adsorption, or through fluid adsorb. So it's not absorb, but adsorb, okay? Which is a holding molecule as a thin film on the outer surface or internal surfaces within the material. And the cells, experts believe, are interacting with this protein layer and not directly with the material surfaces, because the response seems to be pretty consistent. And that we call as a foreign body response or foreign body reaction, which is a unique inflammation response resulting in the formation of foreign body and big cells called giant cells, I will show you later, directly on the polymer surface and subsequent encapsulation of the implant by a fibrous capsule, which is composed of mostly collagen fiber. So let me show you a typical example of biomaterial, which is a surgical suture after like if we had a surgical cut, we need to close it, or from injury. So this is a picture of surgical suture, and the suture, the needle will be removed, but the suture thread will remain into the body. So that will cause a foreign body reaction. And let's take a hemotoxylin and eosin stain picture. So this is a cross-section of the surgical suture. Over time, you can see these sutures. Surrounding the suture, you can see many darker dots, which is represent a nucleus, and some cells having multiple nuclei. You can actually find it over here. And that is, those are called giant cells, which why it's called giant? Because it's a multiple nucleus, not single. Multinucleated foreign body giant cell, we call it. And this is from a macrophage. Multiple macrophages are fused. So one example is this H&E picture. You can see in one single big cell having multiple nuclei. So this cartoon depicts how it forms. So the precursor of macrophage is when they are in the blood plasma, they are called the monocytes. So monocytes are when there is a soft hard tissue and biomaterial causing some inflammatory signal, they will go through from the blood and into the tissue via this chemotaxis from those chemicals signals, and they migrate into the tissue, either soft or hard, to polymer or metal biomaterials. Then at the interface, so monocytes will turn into macrophage, and that biomaterial and tissue interface, they will migrate to that and then try to adhere to the biomaterial, and they differentiate. And somehow the biomaterials are not really eatable, or treatable, so they adhere and differentiate into, they activate the macrophage and try to fuse so that they may try to cover this biomaterial. And so they have this specific giant cell formation. So transition from a blood circulating monocyte to biomaterial and tissue interface, and adherent monocyte macrophage to form foreign body giant cell. So implanted biomaterials and the foreign body response is depicted in this big schematic over time. So when the biomaterials are implanted into the body, like pretty soon, within seconds or within an hour, quickly the biomaterial will be coated by proteins, and that we call protein adsorption. And within 2 days, there will be an acute immune response. So that's the immune cells will interrogate and try to attack this foreign body. The usual innate immunity is coming from neutrophils and then macrophages. And over weeks of the time, now these macrophages are trying to engulf, but they are frustrated. They merge and fuse to form this giant cell formation. And so it can take a week order. And also at the same time, they release a chemical substance such as interferon, interleukin, growth factors, which are secreted by these immune cells and having an effector on the other cells. So to signaling, so that's called a cytokine release. And over weeks of time, over like 3 weeks or longer, they tried to engulf, but they frustrated. So eventually what they do is a nearby fibroblast will secrete collagen to encapsulate this with a collagen capsule, which means if the biomaterials are inside the body, but effectively the body considers it kind of outside of the body by putting this collagen fibrous capsule. So this process can be depicted as a big, a nice figure here. I brought it. So implanted at this bare biomaterial surface will be soon absorbed by protein. And of course, acute inflammatory cells such as neutrophils are getting in. And the matrix deposition with a circulating monocyte gets in, turn them into macrophage, try to engulf it. And then nearby fibroblasts also try to secrete collagen fibers to fibrous encapsulation over like 3 weeks or higher time. And while these foreign body giant cells is formed to try to engulf this biomaterial. So that's the process can be explained as a protein adsorption within seconds to within an hour, recruitment of immune cells such as neutrophils and macrophages from the blood, and these macrophage fusion to form a foreign body giant cells on the order of kind of 2 to 10 days, and involving tissue-specific fibroblasts. Fibroblasts is those who secrete and generate fiber, collagen fibers, and also endothelial cells nearby blood vessel. And subsequent encapsulation of the implant by fibrous capsule. And these are usually avascular. And so here shows a little another like electron micrograph morphology of biomaterial tissue interaction. So SEM is a scanning electron micrograph. So this is more like three-dimensional, pretty high resolution electron micrography showing early thrombotic deposits of now heart-valve material, which as an experiment exposed calf blood for just a few minutes. And it shows that this star mark over here on the surface, you see this undulation. So this shows a protein adsorption. And the B is an experimental tissue reaction to relatively inert biocompatible material. And you show that this material M at the interface of this muscle tissue, you see this thin, very thin, only a few micron thickness, thin fibrous capsule has been formed. And C is similar to the previous one in H&E. I showed you that this dacron, which is biomaterial mesh, and nearby the granulomatous inflammatory response to this surgically implanted dacron surgical suture. So that we call giant cells, and this granuloma as meaning that mass granulation tissue typically produced in response to infection, inflammation, or presence of a foreign substance. Granule means like small grains or rice grains or particles. And if we go back to this scanning electron microscope again, we see these RBCs, and there are platelets, which is a unactivated, and activated seems to have a lot more like branches coming out, and C is a clump. So I brought this picture of an unactivated platelet looks, and once it's activated, you see how striking their shapes are different. So next time we will try to understand the functions of biomaterial, which is involved in open heart surgery. And learning about the medical device, drug eluting stents, now which is using to treat heart diseases. And thank you.